Hello, my name is Jeff Shaw. Thank you for listening. Today I will talk about text mining from journal abstracts. I chose this article by Zaremba. The article was interesting. I think it's important to be able to extract information from information that is already out there. There are many abstracts, many journals already published, all publications that we can already use. It's already out there. And we can use that information and make something else out of it. The authors of this article's motivation was the genome sequencing is very cheap and it can be done anywhere. It's very easy. As a result, there is a plethora of data. It's so overwhelming. But really, this data is almost meaningless. It's just letters and numbers and really worthless. It's meaningless until you assign those different pieces specific roles. And this is called annotation. And that simply is assigning biological roles to portions of a sequence. For example, if you have a gene and you take a section of that gene and you assign it to become a protein, and then maybe a different section of the sequence, you will assign it a role of, an enzyme or something different. So you can see here on the right, at the top is the sequence, and under that is the annotations. So you can judge what the role is of each portion of the sequence. Annotating is very time consuming and it is a brain burning process. It would be nice to automate this process. And that was the goals of the authors who wrote this article. The idea is to analyze the literature that is already there and extract information from the literature and connect that information to genes. And then annotations can become automatic. It would be very helpful, save us a lot of time and a lot of energy. I will talk about information extraction, or IE. The goal of information extraction is to extract helpful information out of a document. IE is a subset of natural language processing. IE is a subset of NLP. We look at natural language, English for an example. We can find entities, and then we can find relations to extract the information to a relational database. Text mining only works if we have the information in a relational database. So that is the purpose of IE, or information extraction. The authors used a program called NetOwl. It was a third-party software. I'll explain a bit how it works later in the presentation. So once the text information was fed into NetOwl, there became an output in the relational database. This is an overview about the process and how it works. It first starts with an abstract from PubMed, and there is so much information. They did not take the full article, simply the abstract, and they fed the abstracts through the NetOwl extraction process, and that process produced entities of interest and relational interests. So that then information went to the relational database. Once in the relational database, you can start the text and data mining process. The picture here is just a diagram of that whole process. The first step is to identify entities. We need to know what we're interested in and what we're looking for. Maybe we're interested in genes and their production roles. Maybe we're interested in enzymes or organisms and their strains, or a variety of different roles that we want to learn more about. First, we have to define what it is we want to know specifically. We also need to think about how to input data. There are many different possibilities and approaches. We could use full articles, abstracts only, old databases, annotations. Really, there's a variety. And the authors chose the abstracts for a variety of reasons. The first reason is that it's free. It's easy to access abstracts of all kinds. Have you seen any journals that block access to abstracts? No, really, that does not happen. So, abstracts are free to access and search for. Abstracts are also compact. They require a low amount of memory and are usually approximately 250 words or less. I mean, if you're working with hundreds and hundreds of abstracts, it's important to have a smaller size because size makes a difference and adds up quickly. So it's best to have abstracts versus using full articles. Secondly, abstracts are very concise and they give you all the information you need right there in that paragraph or two. From a computer's perspective, a full article is full of fluff. I mean, not true for humans. Humans can find useful information, but a computer itself really sees a lot of information, and it's hard to parse out the important information needed from the fluff. So abstracts are nice, concise, easy to work with. The articles took 465 abstracts from PubMed, 237 abstracts were put into the training set, and 138 abstracts were put in the blind set. I will explain both a little bit more later. Step 3 is markup. First is humans, not computers, humans have to read through all of the abstracts with a fine comb and mark up entities of interest. Later, they will evaluate the purpose. There are already predefined guidelines for the markup. And those guidelines were created by computational linguists. So, suppose a biologist will mark up an abstract and then another biologist will review the markups from the first biologist and make sure that they did it correctly. That this is just for the training set and testing purposes. Training and testing purposes. So that's how they know what a true positive looks like. Now to discuss entity extraction. You'll have input, which is, and then output also from the abstracts. We need to know what we want out of the abstracts and from the extraction, so we take the entities themselves. There are a few extraction rules for this process. Now, these are generalized rules based on lexicon, syntax, and domain. These are iterative. You first start with a simple rule and build upon the simple rule up to more complex rules. First, we have to find out, oh, okay, that's an actual name, and then identify that it's a name. And then this name is close to name A is close to name B, and we figure out how they relate and we'll bring them together, and that's how they build upon themselves. You have to start with a simple rule first. If you're familiar with programming, it's very similar to regular expressions, but it's a more abstract process. On the right, you'll see different examples here. The first example and the first step of entity extraction is prior knowledge. For example, using a dictionary, we already know what E. coli is. That's the name of it. You know, same as protein names. We know a specific name of a protein. But what if there are some names that are not in the dictionary? We then have to take advantage of naming convention exploits. For example, gene names typically follow a commonality with an uppercase L and uppercase L. Like an uppercase H, lowercase I, lowercase L, and an uppercase A. If you see something that follows that pattern, you can know that it's likely the name of a gene. Then you can build upon those rules with appositives, verb phrases, and it's very context sensitive. So if one word is close to another word and you pull them together, you may see that they have a relationship. Now, relational extraction is very complex. The entities must first be identified appropriately. Across long text span, it's more computationally expensive. And there are 3 different types. The gene roles, mutant genes and phenotypes, and then organisms and pathogenesis. The table here on the bottom of the slide, you can see the entities have a few rules, but the relational extractions have many rules. And that's because there are many types of relations versus entities. Many more types of relations. Before we dive into the results, we'll look at the metrics. There are 3 different types of metrics. Recall, which is also known as sensitivity, or that maximizes the true positive. Next is precision, and that will minimize false positives. You can see it right here. And just keep that in mind when we go on to the next slide. Now to discuss the results of this study. The take-home message really is that entity extraction performance is typically better than relational extraction performance. If you'll look at the left-hand side, that is the entity extraction process, and the right side is the extraction, the right side is the relational extraction process. So you can see the entity extraction process, typically performance happens much better than the relational process, relational extraction. This slide is a picture of how the workflow works. For example, you have a gene of interest, and you want to see all the relations under that gene. So you click the gene and then pull up all the relations. That will help the annotations later on. You can just read this slide for yourself if you're interested. It's not really super relevant. And the highlighted items are entities. And this is a workflow also. Once you have the gene of interest, you then can extract the relations, and then you can find maybe help identify different roles. Then you can use those relations to later annotate. You may already know where the gene is in the sequence. And here are my references if you would like to review those. Thank you for listening. Any questions?